Hi everyone. Today we talked about the tour of the cell. Specifically, we talked about the organelles that are involved in both plant and animal cells, and this is um, the PowerPoint that supplements that cell organelle worksheet that is in Google Classroom. So we started by talking about the four fundamental units of life, something that we call the cell theory. The first is that all organisms are made up of cells. The second is that the cell is the simplest collection of matter that can be alive, meaning that one cell alone can stand as a living organism. The third is that cell structure is correlated directly to cell function, and all cells are related by their descent from earlier versions of that cell. So these four things put together are what we now know as cell theory. We're going to talk about microscopes in a lab on microscopes, so we are not going to worry about this part of this PowerPoint right now. So I'm just going to skip through it to get to where we need to be. So we talked about eukaryotic versus prokaryotic cells. Eukaryotic cells are those that have internal structure. Their components are compartmentalized, that are very specific to their function. The basic structural and functional unit of every cell is broken up into one of two types of cells, either prokaryotic or eukaryotic. Only organisms of the domains bacteria and archaea are prokaryotic organisms. The other domain is eukarya, and that consists of protists, fungi, animals, plants, and then everything else is a multicellular organism or a single-celled eukaryotic organism. Prokaryotic cells are very basic, where eukaryotic cells are much more complex. But no matter what type of cell you're looking at, all cells have a plasma membrane, a semi-fluid substance that is called a crystal, chromosomes that carry genes, and ribosomes that are responsible for making proteins. Prokaryotic cells are characterized by having no distinct nucleus. That means that their DNA is in an unbound region in the cytosol, called a nucleoid. There are no membrane-bound organelles, and the cytoplasm is bound by the plasma membrane itself. This is your basic prokaryotic organism, a typical bacterium cell, where you have the cell itself with that nucleoid region where the chromosomes are found, all of that DNA. The plasma membrane and everything else would be the cytosol. These little dots are your ribosomes. Bacterium often have one of two different movement or locomotive operations. So here you can see these hair-like filaments. This is one locomotive operation, and this is another one called a flagella. Eukaryotic cells are much more complex. They are categorized by having DNA in a nucleus that is bound by a membrane. They have membrane-bound organelles, and the cytoplasm is the region between the plasma membrane and the nucleus and all other organelles. Eukaryotic cells are generally much larger than prokaryotic cells, and there is a theory that said eukaryotic cells were created when one or more prokaryotic cells were engulfed by each other. The plasma membrane, this is our first organelle that we are going to talk about. This is a selective barrier around the perimeter of a cell that allows for the sufficient passage of oxygen, nutrients, and waste to serve the volume of every cell. This structure is responsible for allowing some things into the cell very easily while making others work a little bit harder for it. The plasma membrane is a protective barrier that helps control substances that are able to enter or exit the cell. This is a general structure of a biological membrane that is considered consistent of a double layer of phospholipids. This is just a cross-section of the cell membrane. We are going to get much more into the cell membrane in chapter 7, so I'm not going to spend a lot of time here. We can skip this stuff. Eukaryotic cells, their genetic information is hosted inside of the nucleus, and then the job of that genetic instruction is carried out by ribosomes. These are probably your two more important organelles in cells. The nucleus is the region of a eukaryotic cell that contains the most of the cell's genes, usually anywhere about 90 to 95% of the cell's DNA is carried in the nucleus. And it is usually the most conspicuous organelle. It's the easiest to see under a microscope. The nuclear envelope encloses the nucleus, separating it from the cytoplasm. The nuclear membrane is a double-layered membrane, much like the plasma membrane, and it consists of a bilipid layer. Here we see what the nucleolus or the nucleus would be this whole region. The nucleolus is a condensed region inside, and then that nuclear envelope, which has pores on it to allow things in and out of the cell. Inside the nucleus, the DNA is organized into discrete units called chromosomes. Each chromosome is composed of a single DNA molecule associated with its proteins. The mixture of DNA and protein is called chromatin, and once it condenses into a rod-like shape, we know those as chromosomes. We will get more into chromosomes, chromatid, chromatin when we actually get into cell division. But the nucleolus is located within the nucleus and is the site of the ribosomal RNA synthesis. Ribosomes are particles that are made of ribosomal RNA and proteins. Their job as an organelle is to carry out protein synthesis, and this happens in two different ways. First, as free ribosomes in the cytosol, and second, on the outside of the endoplasmic reticulum, or inside the nuclear envelope, which is called bound ribosomes. The type of protein they make depends on if they are a free or a bound ribosome. The endomembrane system is consistent of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuole, and the plasma membrane. These components can be continuous with one another or connected via transfer vesicles, which pass materials back and forth within the cell between different organisms. The endoplasmic reticulum accounts for more than half of the total membrane within eukaryotic cells. The endoplasmic reticulum's membrane is continuous with the nuclear envelope, and it has two distinct regions. First being the smooth endoplasmic reticulum, which lacks ribosomes, and the rough endoplasmic reticulum, which is studded with ribosomes. Here we see that rough endoplasmic reticulum studded with those ribosomes and the smooth endoplasmic reticulum out here on the outside. The function of the smooth endoplasmic reticulum is to synthesize lipids, which its main job is to make lipids or fats. It metabolizes carbohydrates, detoxifies drugs and poisons from the cell, and stores calcium ions. Whereas the rough endoplasmic reticulum is studded with those ribosomes, which means that the rough endoplasmic reticulum makes proteins. The Golgi apparatus is consisted of a flattened membrane sacs that are stacked one on top of another. The functions of the Golgi apparatus are to modify the products produced in the endoplasmic reticulum, both lipids and proteins, make certain macromolecules that the cell is unable to take in, and sort and package materials for transport throughout the cell in transport vesicles. Here's our Golgi apparatus right here. A lysosome is a membrane sac of um, enzymes that are responsible for digesting many things in the cell. Lysosomes almost act as a vacuum. Anything that's not supposed to be there is sucked up, digested, and gotten rid of. Worn out cell parts, enzymes that they don't need, bacterias or viruses, anything like that, the lysosome recognizes that it's not supposed to be there, consumes it, digests it, and releases the good stuff back out into the cytoplasm. Lysosomes, much like your stomach, work best in an acidic environment within the inside of the lysosome and maintain that acidity to help digest those macromolecules. We'll come back to that. Vacuoles are in all cell types, but look very different depending on the type of cell that you're looking at. Plant cells or fungal cells may have one large or several medium-sized vacuoles that are derived from the endoplasmic reticulum in the Golgi apparatus. They come in three main different types. First is a food vacuole. This is formed by phagocytosis or cell eating. When the cell senses that there is a food for it outside, it will engulf it in something called a vacuole, a food vacuole, where it will then be brought in, the vacuole will be broken down by lysosomes, and that food will be released out to where it needs to go in the cell. We also have contractual vacuoles. These are found in freshwater protists that's responsible for pumping out excess water from the cell. And central vacuoles, which are found in plant cells, and their job is to help support the interior of the cell by filling itself with organic compounds and mostly water. So here we see that central vacuole, it's taking up about probably a third, sometimes even more of the cell to really help support that cell wall from the outside. The mitochondria, you guys may know this, is the powerhouse of the cell. And although that is a really good nickname for it, it's not actually what the mitochondria does. The mitochondria is actually the site of cellular respiration, which is a metabolic process that uses oxygen to break down food into cellular energy, or ATP. Chloroplasts, which are found in plants and algae, are the site of photosynthesis, the process of taking in sunlight to produce sugar-type molecules. Then we have peroxisomes that are responsible for oxidative organelles, which both cellular respiration and chloroplasts are oxidation reduction reactions. So the peroxisomes really does help with that process. Mitochondria and chloroplasts both have similar qualities. Two different types of bacteria. They're both developed by a double membrane system. Both contain free-floating and circular DNA, and both can grow and reproduce independently of the other cell. These free ribosomes and DNA can only be passed from parent or from mother to offspring. Therefore, you and your siblings have the same mitochondrial DNA, and the only one in your family that would have it different is your father. And that ties into this theory that I mentioned earlier about how there was a theory that early ancestors of the eukaryotic cell engulfed what we now call either the mitochondria or the chloroplast to help create a symbiotic relationship where both are benefiting based off of being merged into one cell. The mitochondria is composed of two matrices, so two membranes. One is the outer membrane with an inner membrane folded into something called a cristae. And then we have that inner membrane space in the mitochondrial matrix. So right here, we see our cristae folded from the inner membrane. That inner membrane matrix is where we'll find the DNA and the ribosomes. And then that inner membrane space is where a lot of our metabolic processes are performed, such as cellular respiration. Chloroplasts have that same two membrane system, except it looks a little bit different. So chloroplasts contain a green pigment called chlorophyll, as well as enzymes that help photosynthesis take place. So that's why for us, leaves look green in the summer. That's because that's where the chloroplast is in the plant and where photosynthesis is happening for that plant. Chloroplasts have a structure inside of them called thylakoids in an internal liquid called a stroma. So here we see a stack of thylakoids, and then that stroma would be that in-between space. Peroxisomes are those specialized metabolic compartments that are bound by a single membrane. Their responsibility is to produce hydrogen peroxide and then convert it to water. A lot of our metabolic processes that happen in cells create hydrogen peroxide as a waste product, and we need to be able to convert that to water because hydrogen peroxide is toxic to cells. These peroxisomes perform reactions with many different functions and have really helped get rid of excess water inside bodies. The cytoskeleton, this is a network of fibers that extend through the cytoplasm. It organizes the cell structure and activities by anchoring many of those organelles to the plasma membrane. It's composed of three different types of molecular structures, one of which is microtubules, second microfilaments, and the third is intermediate filaments. This would visually be the cytoskeleton, so both interior and exterior, where this big blue blob is our nucleus. We're going to jump here. The centrioles and centrioles are only found in plants or in animal cells. And these are actually responsible for controlling the microtubules, especially during animal cell division. Centrioles and centrosomes are responsible for animal cell division. They are controlled by the nucleus when it is time to divide. They move to opposite ends of the cell, stretching their microtubules with them to then move the chromosomes to opposite sides of the cell to create two daughter cells. Here it is visually. They sit at a perpendicular to one another, so one usually is standing up, one is laying down. The whole thing together is a centrosome, and then the centrioles are the two small pieces with microtubules hanging around them. Cilia and flagella, these are your locomotive appendages of some cells. They differ based off of their beating pattern and the shape of them as well. Here we see a flagellum. This is a tail-like whip that makes a whipping motion to cause the bacterium or cell to move that way, whereas the cilia is more of a hair-like structure that moves kind of wavy to help move that cell. The extracellular components depend on what you're looking at. So most of the extracellular components help control communication or strength. There are three main types of extracellular structures that we talk about. The first is cell walls in plants. The second is the extracellular matrix of animal cells and intercellular junctions, which we're not going to get too crazy into. The cell wall is an extracellular structure in plant cells that can help distinguish plant from animal cells. Some prokaryotes and fungi have cell walls, but the cell wall's job for plants is to protect the plant cell, maintain its shape, and prevent the excess uptake of water. They're made of cellulose fibers embedded into other polysaccharides and proteins. These cell walls, they're strong. They create plant cells to have more of a boxy shape to protect the plant cell, maintain shape, pack them tightly together, and prevent the excess uptake of water. Too much water is actually bad for plant cells. So we need to be able to create that balance of what is too much and what is just enough. The extracellular matrix is consisted of different glycoproteins that help with cell communication. It's not very organized, it's a little bit chaotic, but what it does is it actually anchors other cells to each other and allows for communication between cells. The functions of that extracellular matrix is support, adhesion, so cell to cell adhesion, movement, if cells need to move. In animals, we don't want to stand rigid. We want to be able to be flexible and move. So the extracellular matrix helps with that, and it helps regulate to make sure that nutrients are being dispersed around the body evenly. We're not going to get crazy into cell junctions, but cell junctions are how cells communicate between cells of both like and unlike characters. So there's a lot of different types of cell junctions. These are the four most common here, but we're not going to get into those today, which should take us to the end of our PowerPoint. Tomorrow, or our next class, we will be actually drawing the both plant and animal cells so you guys can be able to see visually which organelles actually belong to each other and to which respected cell.